from the University of Wyoming. And prior to that, she did her PhD at Michigan State, and she did a postdoc at the University of St. Andrews in Scotland. In addition to the raccoons, or trash pandas, as some of us like to call them, that she'll be talking about today, she has worked on a lot of different species, including both Asian and African elephants, spotted hyenas, Tasmanian devils, zebra finches, prairie dogs, and lemurs. So, and in the urban context, her group's also looking at skunk and coyote behavior. So super cool work. We're really happy to have Sarah here. And with that, I'll let you take it away.
Thank you. Thank you so much, Sally. And thank you to the rest of the organizing committee for inviting me to give this seminar. I really appreciate it. It's wonderful to be here and to tell you about the work that I've been doing and the work that I am planning to do here at UBC. And it's nice to get introduced to a wider group within forestry. So I'm excited to talk to you about our work, and I'm going to start focused on the work we've been doing with raccoons, and then move into the work that we're planning to do, continuing with raccoons and moving on also with coyotes here in Vancouver. So why are we focused on raccoons? Well, we're really interested in thinking about how wildlife are adapting to urban environments. And that's because urban environments are rapidly expanding across the globe. And as they're doing that, we're increasingly seeing wildlife in our cities. And this has a lot of really important implications, both for the wildlife themselves and also for the people living in cities. And I think that a greater understanding of the behavior, the ecology, and the cognition of urban wildlife is really critical for our ability to maximize coexistence and to minimize conflict with wildlife going forward in urban areas. And cognition hasn't historically been considered too much in terms of thinking about coexistence with wildlife in urban areas, and in terms of how wildlife are able to adapt to and live in urban areas. But I think it's really critical to think about cognition, of course, in conjunction with other factors like life history and diet, in terms of why some species are able to thrive in urban areas while others are not. And that's because is that better? Yeah, okay. Right, and they even need to adjust their communication to cope with increased levels of background noise. And an animal's ability to solve these types of novel and challenging problems in an urban environment can greatly impact its fitness. And there's a hypothesis that I'll be coming back to a lot throughout the talk called the cognitive buffer hypothesis. And this hypothesis posits that the primary adaptive function of an enlarged brain, sorry, I don't have my pointer, of an enlarged brain is to buffer individuals against environmental challenges, specifically by facilitating the construction of novel behavioral responses. And I am going to come back to this hypothesis a lot throughout the talk as a sort of framework for the research that we've been doing. And my students and my colleagues and I have been testing this hypothesis in various ways. And the first aspect of this hypothesis that we've investigated that I'll tell you about today is this often assumed, yet rarely tested connection between bigger brains and enhanced cognitive abilities. So when we think about brain size, we think about how much it can vary across species, right? We see this dramatic variation in brain size across species. And by far and away, the best predictor of brain size is body size. So when you correlate brain size and body size, you see this really nice tight correlation for the most part. Yet there are some species that fall above that correlation, indicating that they're investing relatively more in a very expensive neural tissue, energetically expensive tissue, compared to other species. And then there's other species that fall below this line, indicating that they're investing relatively less in this expensive neural tissue. And so in terms of an adaptationist way of thinking about this, you might think, well, why are some species investing relatively more in this expensive neural tissue? And the answer is likely to be because it has a benefit, right? And when we think about what are the benefits to bigger brains, one of the sort of most obvious answers is that, well, maybe they give, they're associated with more intelligence, right? So bigger brains are likely to lead to higher general cognitive abilities. But as I said, this has been often assumed yet rarely tested. So my colleagues and I set out to test this exact connection. We asked, do species with larger brain sizes, either absolute brain size or brain size relative to body size, do they perform better on a task, a sort of a general intelligence task, than species with smaller brains or smaller brains relative to their body size? And so to do this, we traveled to 9 zoos around the United States, and we tested 153 individuals from the 41 species that are pictured here with a novel problem-solving task. So here is a video. You'll see a series of clips from trials where animals were trying to solve this problem. And so the problem is a puzzle box. So this is a large metal box that the animals have to open to get a food reward. And so to open it, they need to slide like a gate latch in a lateral direction. This allows the door to swing open, and they can then get the food from inside. And so each individual was presented with 3 30-minute trials to see if they could solve this problem. And the food reward was the favorite food of that animal, as told to us by the keeper at that facility. So polar bears get a nice juicy steak, pandas get yummy, yummy bamboo, coatis get hard-boiled eggs, etc. And we had 2 different size boxes to help to accommodate for the variation in the body size of the test subjects. And then we obtained data on adult brain size and adult body mass from the published literature. And we used phylogenetic comparative statistics to assess relationships amongst these measures and performance on the problem-solving task. Okay. So as I mentioned, we tested 153 individuals from 41 species across 9 families within the order Carnivora. And 46 individuals, sorry, you can't see the heading, but I'll tell you, 46 individuals from 23 species across 8 families were successful. And the proportion of individuals within each species that succeeded at opening the box varied considerably amongst families. So species within the family Ursidae, so your bears, were the most successful, followed by Procyon, so that's your ring-tailed coati, kinkajou, and also raccoons, although we didn't specifically test them in this study. And then those are the most successful families, and then the Ursidae, your meerkats and the mongooses, those were the least successful. So as I mentioned, we used Bayesian phylogenetic generalized linear mixed effects models based on a Markov chain Monte Carlo algorithm to identify the variables best sort of predicting success or failure at solving this problem. And so these models allowed us to assess the effects of our predictor variables on success while controlling for shared phylogenetic history. And we found that individuals from carnivore species with both larger absolute brain volume and also larger brain volume relative to body size were more successful at solving the problem. But only relative brain size was a significant predictor of success. And this is because in our individual-based models, when we look at brain size and success, if we take out body mass, that relationship's no longer significant. Okay, so for that study, overall, we find that we're able to give some critical support for this connection between bigger brains and enhanced domain general cognitive abilities. But it also left us with a lot of questions. So for example, if you can get away from just looking at brain size as an overall measure, but actually look at the brains within a species, if you look at the brains of successful individuals versus unsuccessful individuals, what differentiates them? And can we sort of tease out sort of more complex mechanisms that are driving this relationship? And so to do this, we turn to raccoons. And raccoons are really interesting from a neurobiological perspective for several reasons. One of which is that they have dog-like numbers of neurons in a cat-sized brain. So they have really sort of densely intact brains. And when you look at neuronal density in raccoons, you can see they're more primate-like than carnivore-like. So these figures are adapted from a paper by Arlene Messenger and colleagues, and basically they looked at neuronal density of a number of different species. And the triangles are primate species, the gray circles are mammalian carnivores, and the white circles are artiodactyls, so ungulate species, and the red circle is raccoons. So you can see that raccoons are tending to be more primate-like in terms of their neuronal density than compared to the other carnivore species. And so we got together with some neurobiologist colleagues, and we basically looked at the characteristics of raccoons who were either successful at solving a problem or not so successful at solving a problem. And the problem that we investigated was this multi-access puzzle box. So I'll tell you more about this puzzle box later in the talk, but basically you can open this box in 3 different ways. So we had some raccoons who totally failed to solve any of the solutions. We have others who solved 1 or 2 solutions, and then we had others who solved all 3. So we call the ones who solved all 3 successful problem solvers. The intermediates solved 1 or 2 solutions, and the non-solvers solved 0 solutions. And we looked at their brains. And specifically, we looked at this, their brains using a technique called isotropic fractionation. So this is a technique that was developed by Dr. Susanna Herculano-Huzel and colleagues. And basically, what you can do is take a brain and mix it up with liquid and basically form like a brain soup. Okay, and so you can do this with a whole brain, or you can do it with different sections of a brain. And from that brain soup, which is a homogeneous mixture, you can draw out a sample, and then you can accurately count the number of neurons and the number of non-neuronal cells within that sample. And from that, get a very accurate picture of the neuronal density within either the whole brain or these different brain regions that you're investigating. So we use this isotropic fractionation technique on one of the hemispheres of the raccoons. And specifically, we looked at the right hemisphere for this technique. We focused on 2 different brain regions, the somatosensory cortex and the hippocampus. So the somatosensory cortex is really critical for processing sensory input, right? So heat, pain, touch, things like that. But we don't think it's that critical in terms of higher cognitive functions like problem solving, for example. And then the hippocampus is thought to be a really critical area of the brain in terms of memory and other more sort of advanced cognitive functions. So we also did in the other hemisphere, the left hemisphere, we did some sort of more traditional histological analyses to look not just at neuronal density, but also to look at specific neuron types. And I'll get into that in a second. But in terms of the isotropic fractionation, we expected to find no difference among these groups, the non-solvers, the intermediates, and the solvers in terms of the neuronal density or cell density in their somatosensory cortex. But we did expect to find a difference in their hippocampus. And that is in fact what we found. So the somatosensory cortex results were not significant. I'm not showing this here, but there was no difference amongst the treatment groups. But here are the results for the hippocampus. So you can look at, this is the total nuclei. So neuronal and non-neuronal cells here. And we see that solvers have more of these cells compared to the other groups. And then when you look at neuronal density and non-neuronal cells, we actually didn't see a difference in terms of neuronal density, but we do see a difference in terms of non-neuronal cells like glia, which I think is really interesting. So the solvers have more non-neuronal cells than the non-solvers. And glia are thought to be really helpful in terms of information transfer in the brain. And it's actually thought that this sort of higher ratio of glia to neurons is potentially really sort of notable characteristics of the brains of species that are sort of what we typically think of as like cognitively advanced. So Einstein, for example, had a really high ratio of glia to neurons. And humans have a higher ratio of glia to neurons than other primates. So that's, I think, interesting to think about. Okay, so as I mentioned, in addition to looking at the just like this brain soup, we also wanted to do some more traditional histological analyses to look not just at neuronal density, but actually what cells, what neuron types are there. And one of the neuron types that we focused on were these von Economo neurons, or this fusiform shaped neurons. So these are really elongated neurons that are thought to be really critical for rapid information transfer, especially in large brain species. And they've been found in humans, cetaceans such as dolphins, elephants, the great apes, and also the pygmy hippopotamus. And very recently, they've also been found in raccoons. And so we asked, do we see more of these sort of specialized neuron types in the brains of the solvers versus the non-solvers? And that is, in fact, again, what we found. So we do see evidence that the successful problem solvers have more of these elongated fusiform shaped neurons in their dentate gyrus hilus than the non-solvers. And the dentate gyrus hilus is also a really interesting brain region because it's thought to be one of the few brain regions where we see neurogenesis throughout adulthood. And it's also thought to be really critical in terms of exploring novel environments as a trait. So that's, I think, again, a really exciting finding. Okay, so in this study, I think we were able to help sort of validate these measures of cell number and specific neuron types as sort of complementary measures to just looking at overall brain size, which is what has often been done in this sort of comparative cognition literature. And then we were also able to provide, you know, a link between having more of these cells or having denser having higher cell densities in these critical brain regions, and really linking that to some measures of cognition. Okay, so now we've looked at this connection I said here between bigger brains or other measures of like cognitive complexity in terms of neurobiological characteristics and enhanced domain general cognitive abilities. And so my colleagues and I have also been looking at this hypothesis in other ways, and specifically what I'll talk to you about next is looking at these connections between species that live in new, unusual, or complex environmental conditions like cities, and whether species that are thriving in those conditions do in fact exhibit enhanced domain general cognitive abilities, and specifically, are they exhibiting sort of enhanced abilities to generate new or modified behaviors. So this is where I'm going to take you next. So when I'm talking about generating new or modified behaviors, what I'm really talking about is behavioral flexibility. So behavioral flexibility is this sort of multifaceted cognitive trait that includes things like innovative problem solving. So solving a problem in a new way, or a new problem that you've never seen before. It includes things like learning, social learning. So learning by watching another individual do something. Repeated innovations. So repeated innovations are where you're able to solve a problem in multiple different ways. And also inhibitory control. So inhibitory control is really critical for when the environment changes. Can an animal inhibit something that was working? So stop doing that when it's no longer working and switch to something else. So you can think about a raccoon who's really good at breaking into trash cans, right? It's learned to open the lid, get into the trash. And the owner of this trash can doesn't like that. So they come and they put a brick on top or a bungee cord on top, right? So now the raccoon comes, the environment's changed. This behavior of opening the trash can lid is no longer gonna work. So the raccoon can either keep trying to do that, and when it doesn't work, it can give up. Or if it exhibits repeated innovations and inhibitory control, it will stop trying to flip this up, and it will instead switch to doing something else, like pushing off the brick. Okay, so behavioral flexibility is really about, can an animal change its behavior when the environment changes? And we think that this is really critical for animals that are living in rapidly changing environments, like urban areas. And so again, we turn to raccoons as our model organism for investigating whether or not really successful urban adapter species exhibit high levels of behavioral flexibility. And raccoons are a great system for this work because they're so incredibly successful across a huge range of different ecological conditions. They've rapidly spread throughout most of North America in a number of different habitat types. They've also been introduced in parts of Europe and Asia, and they've successfully established populations in those areas as well. When we think of raccoons, we think of them in the city. They're very comfortable in the city. They're very used to navigating through the city. And they're very used to and very good at getting resources in the city, right? So they're very adept at finding food and shelter and using anthropogenic resources in the city. And because of this, anecdotally, they're often thought of as really smart. They're also thought of as pests, right? But because part of that reason is because they're so good at breaking into things. So when I was first thinking about a system to use for this work, I thought about raccoons and I thought, well, surely there must be so much information on raccoons because they're everywhere, they're adorable, and we think they're really smart, right? But when I actually looked in the literature, we know very little, especially about their behavior and their cognition. So this work originally started when I was at the University of Wyoming. And so I'm going to present to you some of the work that we've done there, and then I'll talk to you more about the work we have planned for UBC. So before I get into the actual work, I just want to highlight the amazing students that have been a part of this work, none of the work I will show you is possible without these amazing students, and of course the raccoons. And so just to give you a flavor of what this project is, what we do, we set out camera traps to first locate the animals. And then once we have some good experimental sites where we know there's a decent number of raccoons in that area, we start trapping using live traps. And so we try to get every individual within an area marked. And so once an animal is in the trap, we immobilize it. We get different biological samples. So we get blood samples. So we're able to look at the genetic relatedness amongst individuals in the population. We get fecal samples. So we're able to look at hormones. We're able to look at diet and diseases from these samples. We get whisker samples so that we can do stable isotope analyses. We can look at diet, how that's changing over time, how many anthropogenic resources individuals are using. We generally look at their body condition, we look at their teeth, and we also insert a pit tag into the animal. So a pit tag is like a microchip that you put in your dog or your cat at the vet. So if you lose them, right, they can scan them and call you. So it's the same technology. We insert it between the scapula of the raccoons, and then this allows us to know who individuals are when they come to our experimental sites. And I'll show you some data we've generated from the pit tags a little bit later. And then of course, as part of this project, because we're working in an urban area, there's a lot of opportunities for outreach. And I think it's a really critical part of our work is to talk to people about what we're doing, educate them about the wildlife that's in their backyard, get people involved in the research. It's been a really large part of our work, and I think a really important one. And then we did in Wyoming also have VHF radio collars on the raccoons. We're able to look at home ranges and track movement. There are a number of issues with using VHF radio collars in urban areas. I don't recommend it. So here at UBC, we're leveling up to GPS collars to get better data. Okay, so there's a few sort of general highlights from this work that I want to just mention, and keep these in your brain because they'll come up again later. But for this Wyoming project, we captured and marked over 200 raccoons. And we generally found that juveniles were in poorer condition than adults. But adult females were in better condition than adult males. And raccoons are really good at learning that they don't like to be trapped and then not coming back. So we see really low trappability across seasons, both for juveniles and for adults. So this is frustrating for us because we're trying to get repeated measures on individuals and add, take away and add collars and things like that. But I think it's also really interesting in terms of thinking about their cognition, that they have this experience, they learn from it, and then they don't come back. There are exceptions, of course. There's like a few raccoons that just would be happy to sit in a trap all day and eat cat food, but they are the exception and not the rule. So we've done a lot of work in the wild population. I'll show you that in a second, but I also want to just say that we've done a fair amount of work in captivity as well. So there's a USDA, US Department of Agriculture, National Wildlife Research Center in Fort Collins, Colorado. And we've been lucky enough to collaborate with them, and they have raccoons in their facility that they're using for their own work on, mostly focused on how to create tasty baits for rabies vaccines, to distribute vaccines broadly in wildlife populations. And so we're able to come in and give these captive individually housed raccoons different puzzles for enrichment for their animals. And then it also allows us to gather cognitive data. So I'm going to first tell you about just a little bit of the work we've done with the captive population, and then I'll tell you a little bit about the work we've done with the wild. So I'm just going to introduce some of these studies very briefly, and then I'll get into the details of a few of the studies next. So in captivity, we were able to do this study called the Aesop's Fable. So this is a task from the comparative cognition literature that was based on the Aesop's Fable, the crow and the pitcher. So a crow comes along, it finds a pitcher with a little bit of water in it. It's very thirsty, but it can't reach the water. The water level is too low. So the crow learns to put stones into the pitcher, which raises the water level until it can reach the water and drink. That's the Aesop's Fable. So comparative cognition researchers have taken this paradigm and turned it into an assessment of sort of causal understanding in animals. And so this has been presented to a bunch of corvids and to human children. And so we presented it to raccoons. And the takeaway message, I don't have time to show you all the data, but the takeaway message is that raccoons don't solve it like corvids or human children. They solve it like raccoons. So they basically find really interesting ways to get around what we're giving them. So basically, once you get the animals to drop stones in and raise the water level, you can give them a series of choice tasks to probe their level of understanding. So one of the choice tasks is objects that float and objects that sink, right? So if the animal understands what's going on, it should pick something that sinks. So we had tennis balls filled with sand, right? Those sunk. And then we had tennis balls filled with air, those float. So we gave them both to the raccoons, expecting them to pick the ones that were filled with sand so they could get their little floating marshmallow reward. No, they just take all the balls or all the stones, dump them all in, play with them repeatedly. And then they take the floating ones and they push them down and they splash the water up the side of the tube and they get the marshmallow that way. And we saw this over and over again. The raccoons are just very playful and they just don't behave like corvids, but they solve the problem in very creative ways. All right, we've also, this is a multi-axis puzzle box. This is the one where we then looked at the brains of the raccoons that had solved this challenge. And I will show you that those results in more detail, but basically it's a way to assess behavioral flexibility. We also have done an assessment of behavioral flexibility using a reversal learning task with captive raccoons, skunks, and coyotes. I'll show you those data briefly as well. And then Hannah Griebling, who's actually a PhD student of mine here at UBC, has done this awesome assessment of whether raccoons explore versus exploit puzzles depending on their difficulty level. So I'll show you those results in a second as well. And then in the wild, we've looked at innovative problem solving and learning with puzzle boxes. We've also sort of adjusted these to look at behavioral flexibility. And we've also used reversal learning in the wild to assess behavioral flexibility. So I'll get into these results in a second. So first, in terms of innovative problem solving in wild raccoons, Lauren Stanton, who was my former PhD student, who's now a postdoc at UC Berkeley, designed this multi-compartment puzzle box. So this is like the puzzle box we gave to the captive zoo animals when we were looking at brain size. But because we're studying raccoons in the wild, we need to be able to generate learning curves for them, even if they just show up one night. So we thought, all right, instead of just having one door that they could open, we'll have multiple. So if an animal comes and it opens 5 doors, we can look at its performance as it gains experience with the task and still hopefully generate a learning curve, even if they don't come back repeatedly. And these experimental sites, as you can see here, have this antenna around them. So the raccoons walk over this antenna and that scans their pit tag. And so it tells us who that animal is, when they show up, and when they leave. And so here's a video of a trial. This is an adult male who has a radio collar, and you can see that he's opening these compartments pretty easily. And all of these compartments are open in the same way. So we do have raccoons that can solve this problem. And so when we actually look at the data, we see that 8 of 35 raccoons solve the problem. So that's about 23% of the animals. And animals that try more things are more successful. So this is something I found originally with hyenas, and then we've now seen repeatedly in different species. But basically, when animals are first confronted with a problem they've never seen before, those animals that try more things, so they bite it, they dig under it, they push it, they lick it, right? Those are the animals that eventually are going to be more successful than an animal who just comes up, tries one thing, and when that doesn't work, tries that one thing again, and then eventually leaves. Even if you control for the amount of time at the box, the animals that are doing more, that are trying more things, are more successful. So we see that in the raccoons as well. We also see that juveniles are more successful. And remember, juveniles had poorer body condition than adults, so they might be more motivated to try to solve these types of problems. And they had higher exploratory diversity scores. And this is something we also see in the literature, where juveniles, especially with primates, tend to be sort of more playful and exploratory than adults. And we also do see that they learn the problem. So this is a learning curve. So this is the time they spend sort of really focused on the problem before they open it. And these are the number of doors that they open. So the first time it takes them, over 100 seconds on average to open their first door. But then as they open more and more doors, they very rapidly become quite fast at opening them. Okay, and then we have some anecdotal evidence of memory. So we have 2 individuals that were tested in subsequent years, who both sort of immediately, when they saw the problem, even after a year, opened it very quickly. So that's something to explore further. Okay, so in these trials, gonna work. There we go. Okay, so in these trials, we have a lot of this type of interaction. So especially with kits, we had a lot of individuals coming in groups. And so this leads us to think about social learning. And so basically what we've been able to do with the pit tag data is generate social networks. So we have the arrival and the departure time for each individual. And so from those data, we can generate the connections between individuals. So if you're not familiar with looking at social networks, you can just take this one here, where the nodes are individuals. The line between them is the connection. And so the darker line means individuals are more strongly connected, which means that we see them together more often than not. If there is no connection between lines like P through H are not directly connected, we did not see them together at an experimental site. And so here, the circles are adults and the squares are juveniles. The red are female and the blue are male. And so you can see here that we have sort of 3 sort of distinct communities of adults, but they're connected by this really strongly connected core of juveniles. And this sort of the structure of this network is, if you look at sort of the network metrics, it's likely to be very conducive to information transfer. And so our next phase with this type of work is to look at how solutions are spreading throughout a population, to see, using like network-based diffusion analyses, to see if that spread of information is being transmitted socially, or if individuals are just learning it via trial and error learning. Okay. All right, so this is back to the multi-axis puzzle box. So this was, we're moving a bit from looking at innovation and learning to looking at this actual sort of ability to change your behavior when the environment changes. And so to do this, we turned to a multi-axis puzzle box for the captive animals. And so the multi-axis puzzle box, as I mentioned, has 3 solution types, right? So we present this to an animal on night 1. And if an animal can open one solution type, so let's say the door, if it can open that at least 3 times to show us that it's really learned it, then we lock it, okay? And we give it again to that animal on night 2. If it fails to do a single solution 3 times, it's out of the study. All right, so the successful animals on night 2 now have their puzzle box back. They're all excited. They go to their solution and it's locked. So now we ask, all right, what will you do, right? Do you keep trying to open that locked solution, or do you change your behavior and open up a different one? And so animals that are able to open a different solution at least 3 times now move on to night 3. Animals that fail to do that are out. And on night 3, we now present them with their puzzle box again, except now the 2 solutions are locked. Thank you. Okay, So now on night 3, there's only one solution available. We ask, can the raccoons again change their behavior and just open that one solution? Okay, so animals that are able to open multiple solutions are exhibiting repeated innovations and inhibitory control and thus behavioral flexibility. Okay, so we see that about a third of the animals fail to solve this problem and 2/3 do solve it. And the ones that solve it all solve multiple solutions. Okay, and when we look at learning, we see that successful behaviors again, that raccoons that try more things again are more successful. So here we have exploratory diversity. So that's the number of behaviors that they exhibit in their first trial. And then we have their probability of solving. So once again, just like we saw with the hyenas and the other raccoons, if you try more things when you're faced with a problem, you're more successful. And then we also see evidence of learning. So here we have trial number along the x-axis and time to solution. So that's how much time they spend on each solution. They become faster at solving them. And exploratory diversity actually decreases across trials. So this tells us you try a lot of things, right? You figure out the solution, but like licking is never helpful. So eventually you discard licking and you maintain the behaviors that are helpful. And so we do see with learning that the number of behaviors goes down over time. Okay, I'm going to jump now to this study by Hannah, exploration versus exploitation. So here we wanted to ask if raccoons sort of adjust their strategies as the solution difficulty changes. So we would expect that if all the solutions are easy, animals will keep exploring. Whereas if the solutions are difficult, they would exploit known solutions more and explore less. And so this is a very similar multi-axis puzzle box. In fact, it's the same frame. But now we don't lock the solutions. We just keep giving it to them and not locking any solutions. And so they could just keep exploiting the same solution over and over, or they could explore as much as they want, right? And so we did this, but we also changed the solution difficulty. So first we had to ask, like, did we design this correctly, right? Are the solutions that we think are hard, are they actually hard for the raccoons? And we find that they are, so that's always affirming, right? So raccoons spend more time trying to open a hard solution than the easy or medium ones. Although eventually, as they gain a lot of experience with it, they become quite good at opening even the hard solutions. And then we do find evidence that raccoons adjust their behavior. So when solutions are easy, they're opening more of them. That's the yellow line on the top. They're opening more solutions in a trial, even though they're only getting one food reward. Raccoons are very playful. They just keep opening multiple solutions, and they're doing that less when it's hard. So we do see this trade-off between exploration and exploitation. Okay, so another way to look at behavioral flexibility, now turning to the wild animals, is that we took that multi-compartment box that we had designed to look at innovation and learning. But now instead of all the doors being opened in the one way, they're opened in 4 different ways. So now we ask, okay, can a raccoon, when it comes across this box, it opens all its favorite solutions, gets its food reward, great. Now what does it do, right? Does it switch and open other solution types? Okay, so we looked at this and we found that 7 of the 31 raccoons solved the problem, and all of the 7 that solved the problem solved it in multiple ways. So it's similar to what we saw in captivity, where you either have animals that are totally unsuccessful or animals that exhibit high levels of behavioral flexibility. Interestingly, even when raccoons were tested alone, they didn't always do the same thing. So it's not like the raccoon always opened just one solution type over and over and then switch to the next one and then the next one, right? So they don't have very consistent patterns. They do show diversity in what they're opening, even when they're alone and when all the solution types are available. However, when there's competition, we actually see the raccoons become more self-similar. So when there's other raccoons around, we see that the focal individual is sort of focusing more on particular solutions over and over again. So competition seems to be sort of constraining their innovation in some ways. Okay, so we see evidence of behavioral flexibility in the raccoons in terms of innovative problem solving, learning, repeated innovations, and inhibitory control. We don't yet have evidence for social learning, but that's something we're actively working on. Okay, so in terms of going back to our cognitive buffer hypothesis, we are seeing support for this hypothesis in a number of different ways. I do also have evidence from hyena work that supports that more innovative individuals have higher fitness. But I don't have time to get into that today, but I'm happy to talk about that with anyone who's interested. Okay, so I am running a little bit short on time, so I am going to jump to reversal learning in wild raccoons. So we wanted to assess behavioral flexibility in wild raccoons in a different way from just using this multi-compartment puzzle box. So we turned to this very classic paradigm in the comparative cognition literature, which is reversal learning. So basically in reversal learning, an animal learns to discriminate between two stimuli. So you have two buttons, right? So there's a right one and a left one. So a raccoon learns, if I press the right button, I always get a food reward. If I press the left one, I don't, right? So there's a rewarded side and an unrewarded side. And then once it learns that, once it reaches a learning criterion, say it makes the right choice, the correct choice, 9 out of 10 times, right? Then we switch it. So now the right side is no longer rewarded, but the left side is. And we ask, how many errors does an animal make? And animals that make fewer errors are showing higher levels of behavioral flexibility. Okay, so we presented this, we have done a study in captivity, presenting this to raccoons and skunks and coyotes. We see some interesting differences across the species. But for now, I want to focus on this wild raccoon study. So here's an example of a trial. So an animal comes into this device. This is a automated cognitive testing pod that we designed. So the animal comes in, it scans the pit tag. It says, oh, well, this is Charlie. Charlie's participated in 5 trials. So we're going to present Charlie with trial number 6 in these stimuli, right? So it's an individualized sort of adaptive testing device. So the animal learns, okay, I press one side, I get a food reward. I press the other side, I don't. We found that we tested 40 raccoons. So they're wild, right? They can come in and test if they want or leave whenever they want. And so we had 40 that actually participated. And we had 27 of them habituate to the device. So in that video, the raccoon's just kind of hanging out, right? But it doesn't, it's not eventually gets scared away and it runs, never actually enters the device. So we had 27 animals that actually habituated to the device and actually learned to press the buttons and get food rewards. We found that juveniles were more likely to habituate to the device. Again, remember they have poor body condition, so maybe they're more motivated, but there was no effective sex on habituation. We had 19 of those 27 animals actually learn the association between pushing a button and getting a reward, and 17 of those completing serial reversals. So serial reversal is when you do one reversal and then you do it again and again and again. And so you can sort of generate a learning curve. And ideally, if an animal is like perfectly learning, it should only make one error and then switch, right? After it sort of learns the rule that once it stops working, I need to switch sides. And we found that they do show learning, so they make fewer errors over time. And interestingly, juveniles are making more errors than adults. So you could think, well, all right, maybe the juveniles aren't as smart as the adults, but this is a juvenile trial that was pretty consistent. So juveniles tend to come in these big groups and they all really like testing. And so they jam their bodies into this device that was only built for one raccoon. And so we have a lot of errors of just like butts pushing the buttons, right? So it leads to messy data. So when we look at captivity, we see these really nice clean learning curves. When we look at the wild individuals over on the top, they're a bit messier. In general, the trend is still that they're learning, but there's things interfering with learning in the wild. And interestingly, we also see that more docile raccoons. So we measure their sort of response to people during trapping. And the raccoons that are sort of more willing to let people approach them and are not giving these angry vocalizations, the more docile ones, they're actually learning better than the more aggressive ones. So I think in terms of the conclusions for this, we do see that raccoons most of them are habituating to the device and learning to complete reversals. But it does appear that energetic demands and sociality can explain why juveniles are more likely to habituate to the testing device than adults, because they might, in their groups, they become sort of less afraid and they're sort of more willing to engage with novelty. And that, you know, we do see that there's sort of messier data in the wild, although they are learning. And we think that this social interference, sort of these like competitions that we see between the juveniles, can be explaining a lot of that messy data, and that it's particularly challenging when testing animals in the wild to deal with things like social interference. So I kind of want to end with thinking about what does this all mean in terms of human-wildlife conflict and coexisting with wildlife species in urban areas. As I mentioned, raccoons get into a lot of trouble with people all the time, and we hear about this all the time when we're out in the field. And so this got us thinking about sort of the cognition of nuisance, so-called nuisance animals. And is it possible that, in fact, while cognition might be enabling some animals to live in urban areas, it's also bringing these sort of smarter animals into the most conflict with people? And alternatively, are sort of really intelligent animals actually able to avoid conflict with people? So we wrote a review paper looking at what we know about the cognition of nuisance animals. But in terms of thinking about our own data and the implications for human-wildlife conflict, I think we have a couple of pretty interesting results. So the first is that juvenile raccoons may be particularly problematic in terms of habituating to a lot of the non-lethal mitigation strategies that we have for dealing with human-wildlife conflict, because they tend to have poor body condition, they're more food motivated, and they tend to habituate to human devices faster than adults. And then also, in terms of thinking about management challenges going forward, maybe it's not the aggressive animals that we have to worry about so much, but actually these more docile individuals that are maybe smarter, maybe they're the ones breaking into the chicken coop and eating your chickens, and they're in the long run potentially going to present more of a management challenge than the sort of bolder, more aggressive animals. Okay, so the last thing I just want to mention quickly is what we're doing here. So we've started the UBC Urban Wildlife Project along with my amazing graduate students, Hannah Griebling and Jaylene Bastos, and we've gotten all of our permits in place. And so we've started setting up our camera trap transects. So we have these transects now going through the city of Vancouver. And we joined the Urban Wildlife Information Network. So this is a large network of labs all over North America and now also in South Africa and Germany that are running similar transects in urban areas. And so we're able to look at the wildlife that we're seeing in urban areas, not just in Vancouver, but also compare that to what's happening in many different cities all over the world. And so we're able to look at sort of broader patterns of what's going on with urban wildlife. And so our next phase now that we have these cameras out is figuring out where are the raccoons in Vancouver, where are the most promising experimental sites, and then we'll be starting our trapping work this summer. And what we're doing, we're really thinking about continuing this work, thinking a lot about cognition, but then also sort of putting it into this integrative framework, looking at how health is influencing cognition and vice versa, and also looking at changes in the patterns of sociality of these urban wildlife, and also really quantifying environmental harshness, both in terms of looking at sort of percent of impervious surface cover, but also looking at other things like human tolerance for wildlife as measures of sort of environmental harshness and complexity. And then the last thing I want to mention is I think we're all familiar with these headlines that we've been seeing over the last year and a half or so about all of these aggressive coyote incidents that were happening in Stanley Park. And unfortunately, it ended up in a call of a number of the coyotes that were living in Stanley Park. And I've been approached to help collect data on the coyotes in and around Stanley Park. And so that's another program that I'm embarking on. So hopefully the funding will come through with that. And I'm collaborating with Bill Harrower from the BC Ministry of Forests, also other faculty at UBC, including Cole Burton, Saliato, and Kristen Walker, collaborator at University of Alberta, and also the Stanley Park Ecological Society and the Vancouver Parks Board. And so we're embarking, hopefully, on this really big project to collect data on what's going on with the coyotes in Stanley Park and other areas, and then how can we sort of come up with really informed management solutions for this population in the future. So with that, I just want to thank you for listening to this talk. Hopefully, I have convinced you that a greater understanding of the behavior, the cognition, and the ecology of urban wildlife is really critical for our ability to coexist with wildlife going forward. Of course, there's many people to acknowledge, in addition to the raccoons as well. And with that, I'm happy to take any questions in the time remaining.
Okay, thanks very much, Sarah. And I am going to stay here so I can take questions online. Super interesting talk. And the first question that we have here is from Kathy Martin, who's one of our faculty members, and she is watching from PEI, I believe. And she asks, there are evident benefits to being a solver. Are there any costs to being a solver? Is it always better to be a solver, or is there a cost benefit trade-off operating?
Yeah, that's a great question. Thank you, Kathy. Yes, so in the study I briefly alluded to, where we looked at the fitness consequences of innovation in hyenas, we were able to do that because it's a really long-term field study. And so I collected data on innovation in hyenas for my dissertation, and we now have data on the sort of reproductive success of the females that participated in my study and what's going on with their offspring. And so from that, we're seeing some really interesting trade-offs where innovative females are producing more young. So they're having larger, they're having like more frequent litters, but there's lower survival. And so I think that there is definitely a trade-off where they're able to gain access to more resources. And that can obviously have benefits in terms of producing more offspring, and being able to feed those offspring. And those offspring are surviving for the first couple of years pretty well. But then we do see this decline in overall offspring survival. And it's likely that because those innovative females are potentially bolder, more curious, more exploratory, right? Maybe they're getting into trouble. Maybe they're getting coming into contact with humans, and that leads to problems like, or getting poisoned or running into lions, things like that. So I do definitely think there are trade-offs. Yeah.
Interesting. Any questions from the room? I have another, oh, Rob.
Maybe more along the lines of the selection part of your model, but is there any difference or any evidence out there in any species for, say, differences in neuron density or glial abundance, or, say, fusiform neurons that you were talking about between city mice and country mice, or city raccoons?
It's a great question. I don't know off the top of my head of any studies that have looked at that. Specifically, this isotropic fractionation technique looking at neuronal density is relatively new. So they've done these great comparative studies looking across different species, but they haven't done any, as far as I'm aware, I don't know of any studies looking at urban versus rural. I also don't know of any looking at other sort of neurobiological characteristics. It's really hard to get those samples. Like I put out a call, like if you happen across a dead raccoon, please let us know. And by the time we got any raccoons, they were just too old to actually be able to collect the brains. So this study we were able to do because they're in this captive facility. So they were tested, and then they were put down as part of the USDA's other studies. So we said, all right, since you're putting them down anyway, like we'll look at their brains. But yeah, trying to collect the brains of actually wild, urban and rural animals has at least proven to me to be too logistically challenging, unless you have a lot of people that are like really on top of it and can get you these carcasses very quickly. Yeah.
So we've got one more question online, and this is from Estefania Milia Moreno, and she and there's also many thank yous and comments about what a great talk it was. But Estefania asks, many raccoons are building nests on roofs of UBC family housing, and the residents have been told they are protected by law and can't be relocated. Any suggestions for coexisting with them?
Yeah, so we get this complaint a lot. Basically, our suggestion is wait until breeding season is over. Wait until the kits have moved, become independent, left the nest or the den. And then we try to board up. So a lot of the complaints we get are raccoons that have actually gotten into attics. and things, right? They found like a hole or in a chimney. And so the key thing is just to block access. So once the raccoons have left, you sort of board it up and then they can't come back. But you have to wait until the kids are independent, obviously, or it can lead to bigger problems. So that's our biggest recommendation, but it's a challenge, definitely. And it obviously, if they are somewhere just making a bunch of noise where you can't block it off, then that's a different problem entirely. Yeah.
Right. So I had a question for you. And so this whole realm of problem solving and play and the more adventuresome behavior, I guess, that you've observed in some animals and not others, there must be parallels in studies of children's behavior and problem solving. I always wonder if you're tempted to take the puzzle boxes home, but anyways, but are there parallels with studying human behavior and sort of different axes of behavior, which relates back to Kathy's questions and so.
Yeah. There's obviously a huge literature on human development and cognitive development in children. And I have taken, for example, the exploitation study I specifically took to a child like development specialist. I was like, do you know, because we couldn't find any similar studies in the animal cognition literature. And I said, well, surely there has to be studies like this in human children. And we actually couldn't find any, but we did start talking to people in that world, and it's really interesting. And I really like, when I can, and whenever I have free time, I really like sort of watching lectures from that field and reading papers from that field, because I do think it's I do think there are a lot of parallels, and I also have young children, so I see what they're doing. And I do think there's definitely, especially in terms of play and exploration and creativity, curiosity, a lot of those characteristics we see in raccoons. And I definitely think they're really critical in terms of how human children are developing and gaining cognitive skills. But it's a great question. And we haven't had like a formal collaboration yet with people from that field, but it would be wonderful to do that. Yeah.
Okay, well, thank you again for a great seminar, and please join me in thanking Sarah one more time.